to the judges. My name is Vikram. My presentation was based on modeling alternative bacteriophage therapy against multi-drug resistant bacteria using ordinary differential equations. First off, the main need for this is the global crisis of uh, antibiotic resistant bacteria and multi-drug resistance bacteria. And multi-drug resistant bacteria, which are like resistant to multiple antibiotic classes, they're one of the biggest public health crises for the reasons of harder to treat infections. They have increased patient mortality, higher costs, and extended hospital stays. And it's been released into multiple communities, so it's not just limited to one specific area. It's appearing all over the world, everywhere you go. In 2019 alone, which is almost like a decade ago, um, MDR bacteria were associated with roughly 1.27 million deaths worldwide, and by 2050, it's projected to reach about 10 million. Should the problem, I put problem go unchecked, but now it's more accurate to say that if the current rate of discovery for antibiotics stays constant. The main culprits of MDR cases and MDR deaths are the increasingly hard to treat escape pathogens, which are six pathogens known for their diverse resistance to antibiotics and their incredibly high mortality rate, with mortality rates ranging from 20% to upper 50s. Why are the antibiotics failing? But more or less, they the escape pathogens have they gain resistance really quickly, and they're resistant to last resort antibiotics. So the antibiotic pipeline is really drying up. And due to the biological mechanism of horizontal gene transfer, they are able to, bacteria species of any kind can and do frequently share genes with one another. And it leads to widespread antibiotic resistance, say like the E and escape got resistance to the bacteria and the patient were to die, it's very likely that in a very short amount of time, all the other pathogens of the sort would also gain resistance to that antibiotic. My study focuses on bacteriophages. Bacteriophages are they're just viruses, and they kill bacteria. They don't harm the human cells. Phages, being viruses, they adapt and evolve alongside bacteria, and it's maintaining effectiveness. They don't antibiotics right now, they just work in one specific way, but the adaptability of phages makes it much better because they are able to still lock on and kill their bacteria target, effectively all the time. Antibiotics indiscriminately attack all organisms with the biological processes that they're designed to disrupt. And this is a problem because it's like carpet bombing. It harms the bacteria that is hurting us, but it also harms the good bacteria in our body, which causes a bunch of problems. But phages are the leading form of treatment because they'll only seek out, it's like a specific missile. They'll only seek out the bacteria that they are, like, I guess, biologically programmed to reproduce in. My hypothesis is using ODE-based modeling, we'll see that bacteriophage cocktail treatment will significantly reduce the growth rate of multi-drug resistant bacteria in a liquid culture compared to antibiotic-treated MDRs. These are my methods. My conceptual framework is to just more or less just recognize the mechanism of bacteriophage therapy before I create my differential equation. It's This is not like the newest figures of what am I saying? This is not old news. Viruses, bacteriophages, they all work by having genetic material attaching themselves into a host cell organism. They hijack their bacterial machinery through the nucleus, and they destroy the host, release a bunch more bacteriophages, and the cycle continually repeats. This is my literature validation, more or less literature studies and reviews showing that this is a very viable form of treatment. And MBio study from 2021, the biggest study in my project. The phage cocktails were able to rescue mice from lethal infections caused by the K in escape Klebsiella pneumoniae. And additional studies report that personalized phage treatments clear MDRs like the A in escape and the infections that antibiotics would just have failed to control. Mathematical modeling also substantiates these findings as ODE systems illustrate the predictive capabilities of absorption rates, burst size, and decay terms regarding bacterial clearance. Collectively, laboratory data and clinical trials will be able to more or less accentuate the viability of phages as a critical approach to addressing MDRs. The ODE I created to simulate the phage cocktail um, was a growth logistic model for the susceptible bacteria, and then the infected and free phage bacteria were just based on simple addition and subtraction. Um, the This is the the growth logistic model, and this is more or less that's the negative part of it is the minusing of the free phages and the susceptible bacteria getting infected. So this is the infection, and that's the growth logistic model of the just free bacteria. The differential for the infected bacteria is the infected bacteria minus bacteria that is then killed from the lysis rate. And the differential for the free phages is the infected bacteria minus the decay rate of the free phages minus the amount of phages that's going into susceptible bacteria. My workflow, I guess the steps in which my study would take its course would be for the MDR, start MDR bacteria detected in my liquid culture model. The baseline control is the untreated growth, no phage 0 = 0, and intervention arms, which is my single phage cocktail, and phage plus antibiotic. The ODE simulation is an SIP model that I used RK4 integration tools for, found on Replit. And the outputs are my amount of life between a 12-hour time interval over a 48 period. These are my input values for for the susceptible, all the variables. All the typical ranges that I found were from literature studies that I've put in my references, and I more or less chose the middle value for all of these as a easily testable model. After running them through RK4 integration tools that I found online within coding spaces like Replit and Mat tools, I was able to glean these results, which is at the beginning, you start with a bunch of free bacteria at time 0, and afterward, there's no free bacteria and they're all just infected. There's the amount of free phages keep increasing, and the amount of infected bacteria very, very quickly diminishes compared to the antibiotic only treatment, which is where the amount of bacteria dwindles, but is not completely gone. It only halves versus a complete annihilation in less than 24 hours. Um, the side-by-side data really just illustrates the difference in mechanisms. Antibiotics act more linearly, with a fixed potency that can be resisted, and it's not recommended to use antibiotics as much anymore because it's a very high very high, uh, I'm losing my words, very high chance that the bacteria is going to gain resistance, whereas phages are able to adapt and quickly kill off all the bacteria that they are not trained, that they are programmed to kill. I didn't add that. Um, Really, the limitations is the fact that it was done on a mathematical model instead of a simple mathematical model. There wasn't really an immune system compartment. There wasn't an explicit resistance strain, and it wasn't done in an in-person lab. So to reach a more accurate, to reach the most accurate model I could possibly have is just to do in a laboratory, but to reach a more accurate model for ODEs, I simply need more variables, which I was not able to find in literature review. It's my references, and this is the references um, for the variables.